So hello everyone, thank you for joining today's session. We'll be starting with the session in a few more minutes. We are waiting for the rest of the participants to join. All right, I can see that few participants have joined. So hello Dinesh, hello James, hello Olu Vasigan, hello Dr. Ramesh. We'll be starting with the session in 4 minutes. And meanwhile, we are waiting. It would be great if you could introduce yourself in the chat box about your educational background and research interest. All right, so Dinesh says his background is in chemistry and his research interest is in drug discovery. All right, can you also let us know if you're joining with us for the first time or have you previously attended our Cheminformatics webinar? All right, it's your first time. So you're yet to enroll for the program, right? And what about the rest of the participants? We'll be starting in a few more minutes. So it would be great if the participants can introduce yourself in the chat box, just like Dinesh, so that we can get to know if you're joining for the first time or have you enrolled for the program. All right, Neda is joining for the very first time, and Dr. Sara is from a microbiology and molecular genetics background. I'm curious to know about the rest of you as well. Sanobar says he's enrolled for the program. Perfect. And Ahmed is from a bioinformatics and computational biology background. All right. And James Ogidi is a pharmacist and has research interest in discovery of drugs for neurodegenerative disease therapy. And Ole Vesugan is from a pharmacy background as well and has interest in discovering anti-tumor agents and have enrolled for the program. Perfect. Mahanaz is yet to enroll for the program and would like to know about the live sessions of this program. And Sufyan is also coming here for the first time and is from molecular biology background. And James has already enrolled for the program. Perfect. So we have a mix of participants who are enrolled and who are yet to enroll. So meanwhile, the participants are introducing themselves in the chat box. I'll start with today's session. So hello, everyone. A very warm greeting to one and all. My name is Srigauri Krishnakumar, and I would like to welcome you all for the very first session of the Cheminformatics for Biomedical Drug Discovery Program. This program is designed for beginners and students who are eager to learn about the fascinating field of computational structural biology and cheminformatics. And in today's session, we are joined by our program mentor, Dr. Puneet Kakkar, who will cover several topics on molecular structure of biological macromolecules, protein structures, recent advancements in drug discovery, protein-protein interactions, biomolecular visualization, among several other topics. So before we begin with today's session, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. And as a part of the program, students will have access to the omics logic training that has been completed by over 24,000 participants from 187 countries in over 300 workshops. And this training has been completed by participants in 6 different specialization tracks that include oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data. And students will also have access to the omics logic resources, which takes a combination of training modules, data analysis tools, curated project data sets, and interactive sessions with mentors to give the student a clear path of the Bloom's taxonomy pyramid. And this also comes with an access to the omics logic courses, which include courses on bioinformatics, precision medicine, multi-omics that consist of genomics, transcriptomics, metagenomics, and epigenomics, data science that includes bioinformatics using R and Python, as well as cheminformatics, and example projects of different fields which you could learn from and replicate for your research work. And users will also be given directions to access the T-Bioinfo platform for bioinformatic processing and analysis of data. The platform includes demo pipeline, as well as data management and analysis cloud infrastructure to run the bioinformatic pipeline. And different stages of analysis are performed on different sections on this multi-omics platform. So now to view the various courses and example projects, student projects, you need to first sign up on the Omics Logic Learn portal. I can see that several of the participants are joining us for the first time. So let me walk you through a brief demo on how you can sign up. So I'm sharing the link to the Learn portal in the chat box. So please visit the link and I'll walk you through how you can sign up. So when you visit the link, you'll be able to see an option that says create an account. When you click on that, you will be able to either log in, sign up using by adding your name, email ID, and password, or you can sign up using any of your social media apps. This could be Google, Facebook account, Apple account, GitHub account, or even Twitter. And for those participants who have already enrolled for the program and who already have an account on the portal, you can simply click on the login now option that you see here and click on sign. So I'd like to request the participants to quickly let me know in the chat box if you have signed up or not. If you have signed up, please put a done. And if you are yet to sign up, once you have signed up, please put a yes so that I know I can proceed further. And if you are facing any kind of technical issues, you can share them as well in the chat box, and I'll be happy to answer. Mahanas has signed up. And what about the rest of you? All right, so I can see that participants are signing up. So I'll proceed along so that if the participants have any queries, you can let me know in the chat as well. So since I already have an account on the portal, I'm going to click on sign in. And once you sign in, you will be able to see the various courses and example projects that are there on the portal. So you can simply enter the keyword that you're interested in the search bar that says looking for a specific course. So for instance, Cheminformatics, and that will show you the courses that is associated with Cheminformatics. And now the most important part for the participants who have enrolled for the program, as well as the participants who are yet to enroll, you need to update your account on the portal. For that, you can click on the tab that says welcome back with your email ID. And once you click here, you will be able to see your profile settings. When you come here, you'll be able to see an icon that says profile. Click on that, and that will enable you to edit all the account settings. And some of the important features that you need to update on your account is, first is that you need to enter your name and make sure that it is in proper capitalization. And you need to upload a profile image for yourself, as well as you have to add a bio about your that will consist of your education background and research interest. And finally, you can link your social media handles as well. And now you'll also see various tabs in your profile section, that is activity, wherein the community manager and the program mentor will be tracking your learning progress. Under the courses tab, you'll be able to see the various courses that you're currently completing. And under the programs tab, you'll be able to see the program progress that you're making once you enroll for any of the OMUX project programs. And finally, after the completion of your coursework and associated assignments, the certificates will appear under the certificates tab. So that was all about signing up and navigating through the portal. So if the participants have no further queries, I'll pass on the stage to the program mentor who's starting with today's session. But along the way, if the participants have any queries, please feel free to share it in the chat and we'll be happy to answer. So the stage is all yours, Dr. Puneet Patel.
Thank you very much. I hope you are able to see. I hope you are able to hear me loud and clear.
Yes, sir, you're perfect.
Very good. Let me share my screen. Okay, screen, the whole. Okay, let me see it. Yes. Are you able to see my screen?
Yes, sir. The slideshow is showing up. Yes, and now it is visible.
Very good. Let me talk it to the top. Do this one more. There is an option. Yes, turn on the laser pointer. Okay. Yeah. Okay. All right. So thank you very much for joining today's session. It is more on the structural biology side. We will cover some of the basic stuff in the biomacromolecule side, first of all, so that when we go a little complex in the upcoming session, you are well acquainted with what we are talking, why are we talking about those things. And these are very, very basic. Maybe you are already aware of it, but you will need to see a little bit of different perspective of how we are seeing the cell, how are we seeing the porous cell membrane, how are we seeing the structure of a DNA or a protein, and likewise. Let me begin with the presentation outline. We'll talk about a little bit on the cell, the unit of life. I know most of you are very much familiar, but there will be a certain aspect of it that you will connect at the fifth point of this slide. Then we'll talk about the central dogma, which is something very briefly we'll discuss about the flow of information. Why are we talking about it? You'll understand. Biomacromolecules, DNA and protein, some bit of their structure, so that in case you need a quick recap of what and how they are connected, that should be covered here. Then we'll go to the exercise on DNA and protein sequence and structures. And then we'll talk about protein-protein interaction, and together how you can connect the cell and how the drug discovery campaigns start, and you can visualize those things in the whole process. So I will not take much time, just will move forward, and I hope you will enjoy the session. The whole session will be pretty much covered in several hands-on activities, so be with me. All right, first thing first, we are talking about the cell. We are talking about the basic unit of structure of life, right? We are saying the animal cell is something which is very fundamental. Whenever we are talking about the drug discovery, we have to understand, when we are talking about a medicine, that we take it via oral or the intravenous route, where it goes, how it works, where it interacts, what are the key parameters that are responsible for it. That is something which is very, very far from here. We have to cover so many milestones in between. But the overall fundamental is, we should know what are the constituents of the cell, what happens within the cell environment, what happens outside the cell environment, what making the difference between internal and external region of the cell, how the cell communicates internally and externally, and many more things. Important thing that we need to understand, that our body is made up of cells, billions of cells actually. And the cell, we are saying, which is represented here, that you have studied in your school education, it's lot many things. Cells are of different types. Cells are primitive, cells are advanced, animal or plant cells, eukaryotic cells, primitive like prokaryotic cells. There is a basic difference in animal and plant cells. Some organelles, subcellular parts are different. Chloroplast, for example, present in plant cells but not in human. Very well defined nucleus present in eukaryotic cell but not in prokaryotic cells. Many other components which are responsible for certain specific type of function. All things are very, very important. In general, the cell can have three main critical parts. One is the cell membrane, which covers the cell, protects it from the outside environment, so that nothing like everything is going inside and anything from the inside is going outside. It's very much regulated. Second is the cytoplasm side, where all the cell organelles are basically swimming around. And the third part is the well-defined nucleus, which has its own nuclear membrane, and inside that is a chromatin or chromosome, which is present, which regulates the flow of information. So the membrane of the cell seems very smooth, but it is not actually. It has pores. The fluid mosaic model, if you have studied in the past, you should revise that one, where it is made up of lipids and proteins. Proteins are floating around, and membrane is like, has a capacity to include proteins which are having the flanking region outside the cell environment, outside. So everything is outside the cell, outside the cell membrane is the extracellular environment. Anything which is inside this is an intracellular environment. So it is important to differentiate intracellular and extracellular environment, and you will see in the last slide why are we saying this. Now, body is made up of multiple organs, organ system. Organ system, if you go a little bit down, you take out one organ which is made up of tissues, multiple types of tissues, and a tissue is made up of cells, in fact. So cell is something which is basic structure that defines the life, and it is important that constituents should be very much structured, and we should know about them. In general, some structures, some cellular uh, parts of the cells are like mitochondria, kind of powerhouse. You have rough endoplasmic reticulum, which has the endoplasmic reticulum and encoded with the, this the endoplasmic reticulum is embedded with kind of ribosomes for protein synthesis. You have some vacuoles, you have Golgi vesicles, Golgi apparatus, which defines the fate of the products, protein products, where they should go, where should be targeted, whether they are going to the cell membrane, they are going extracellular space, they are going to any other place, remain in cytoplasm, and nucleus and the chromatin and the DNA, which is present, transcribe, translate into protein products, which are responsible for basically messaging and providing necessary function within the cell. So in short, cell has many things. There is a division of labor kind of thing where organelles has their own defined functions, very critical, and there are a lot many things needs to be understood in cells, so that the concept of drug discovery delivery should be little bit understand clearly, or should be understand clearly. Now when we are saying we are taking any medicine, it goes into the stomach and intestine, then blood, then it has to go across the whole body. Then with the help of blood, it has to reach to individual cells where the blood supplies are also there, right? Because they are also receiving nutrients, food from the blood. Now, when you are taking any medicine, the medicine also delivered to a place where the cells are ready to take up the medicine. So medicine has to reach to a place. Now, sometimes medicine has some specific target. We'll talk about those targets. If you don't know what is the meaning of target, don't worry about it. But understand that there are some macromolecules, biomacromolecules, which are important for um, certain function, so that if their function is modified, the disease can be cured. Now the drug molecule has to reach to the place where the target is present. Now, if the target is on the cell membrane, it has to bind to the target, which is on the cell membrane, and should trigger the function, necessary function. But what if the target is something inside the cytoplasm? It means that your drug molecule has to penetrate, go inside the cellular environment and reach to the target in a sufficient quantity, or wherever the requirement number of molecules are required, basis the expression of the particular target. Sometimes targets are located on the nuclear membrane, or the DNA can be also a target. In such cases, drug has to cross all the barriers, right from the um, oral cavity or intestine, liver, kidney, blood, and then reach to the cell membranes, cross the cell membranes, all travel through till the receptor place in the nuclear membrane, and then bind it. Now you have some idea what we are talking about. We are talking about something at a level of cellular structure. We are saying that even if your aim is to make a molecule which can act as a drug, it means that you are looking for something where the drug should go and bind. Where these targets are present, these targets are nothing but they are present nowhere but on the cells or the cell surface. Something I believe you are with me. Let me go to the next level. We are talking about the cell. We are saying the cell is made up of multiple things, and we also said that there are many macromolecules which acts on the product made by the cell. Okay, all right, products made by cell. What are those products? These products are proteins, macromolecules. Mostly the proteins are the targets, and very important topic of today's discussion as well. How the proteins are made? The proteins are made basis what is the flow. We call it as flow of genetic information which passes from DNA to RNA and on the protein. If you see the left hand side image, we say that DNA, which is present where in the nucleus, it transcribes into the RNA. RNA goes and produces the protein in the cytoplasm. Now this protein is produced in cytoplasm, but it has to be targeted also, right? It has to be targeted on the cell membrane, it has to be targeted on nuclear membrane, on the cytoplasm. So all the fate or maybe extracellular environment of the cell. So that specific suborganelles organelles of the cell functions that way. But important here is the proteins is produced from the very regulated, controlled fashion, very foolproof method. After a lot of proofreading, a correct, accurate protein has to be made for certain specific functions. Okay, all right. But is it that simple? No, actually, each and everything is very, very complex here. Just a little bit of sophistication is shown on the right-hand side image, where you see the DNA portion, which is making the RNA, which is giving the information that what has to be transcribed, which protein has to be produced, has to uncoil and should open the strand so that the RNA can be made for that specific region. But DNA is highly coiled, super coiled in forms of the chromosome. When you um, when you when you do the I would say when you loosen the chromosome, you get the chromatin fiber. Chromatin fiber is still coiled on the condensed chromatin, which are basically coiled over the histone proteins. And then if you uncoil them, you get the double helical structure of the DNA. Some part of the DNA should open up, create the mRNA, coiled it again and form this whole function, the whole structure. Now this part, for example, is going to produce the mRNA, which is created here, and then RNA is used to produce the protein. So in short, all the information of what has to be produced is encoded in the form of a chromosome, in the form of a DNA or gene, and that is in turn present in chromosome. Cell is busy in producing so many protein products inside the cell all the time. That's why it's a very busy machinery. You can say it's a very busy machinery. Now, what is important is in the whole process of manufacturing the proteins, there are some mistakes also happens. There are many mutations. We call it as the modification of the amino acids, or modification in the DNA happens because of um, many effect on the environment, present in the environment, or we are exposed to during our development time. These causes the modifications, and these modification leads to a different protein. Now, if the protein is changed, the function is also going to be impacted because it's a highly regulated process. And the structure is also very much conserved so that it can do a good job for its basis, its structure. You will see those things in the coming slides. But point is that when there are certain changes happens in the product, the changes leads to certain diseases. Many times it leads to a certain disease because the protein is not functional. Protein which is produced is not good quality, it is not doing its job correctly, or it is extra produced, it shouldn't be produced, but it has somehow produced, or it has an added functionality which is not required in the body, for example. In such cases, we see there is a disease. Because there is a disease, there is a problem is has to be solved. So we have a drug discovery in mind. That's why people use external molecules, uh, different therapeutic strategies to somehow rectify or correct the function, or somehow modify the process, so that natural process should be should be should be preserved into the system, and the disease and the disease should be cured, or we can stop the progression of the disease. But in all the cases are not possible. There are so many diseases. All right, we said that the process of from DNA to RNA, RNA to protein is there, but you can ask me why, how the DNA looks like. Now, you have also studied that part, so everything is very redundant maybe for you, but now the story is that you need to see those things from the structural point of view. And that's the topic for today's discussion. When we say the structural point of view is that you should know how they are actually in reality look like. And when we say that, okay, DNA is made up of base pairs, which are nothing but 4 ATGC, adenine, thymine, cytosine, and guanine. G pairs with C, A pairs with T, double bond, 2 hydrogen bonds, 3 hydrogen bonds, and like that. You see in the left hand side image, you have adenine, thymine, you have hydrogen bonds here, and you have guanine, you have cytosine, and you have hydrogen bonds here. But exactly how they look like, we have a protein data bank entry. I will tell you how to search it, because it's a very well-known biomacromolecule structure database, and it's a very key initial point for you to go for the rational drug design. But before that, understand that DNA is a double helical structure. It has 4 bases: adenine, thymine, guanine, cytosine. This is how they interact, and the pairing is important. If the pairing is disrupted, it may not either uncoil, may not be broken by the protein enzyme. If it is broken, then it cannot be uh, joined again, and it may cause problem. And because such cases are known, the drug discovery also happened that way, that we design molecule that binds to the DNA. On the right hand side, you have a protein. Now, protein has multiple level of structures. We call it as primary structure, secondary, tertiary, and quaternary. But it's very fundamental, it's very important. We should understand what do they actually mean by doing some hands-on exercise. We are saying the primary structure of the protein is nothing but the sequence of the protein. And we will talk so many times about the protein sequence and structure in the upcoming sessions as well. The amino acid names are encoded by 3-letter code here: asparagine, glycine, phenylalanine, glutamate, something like that for all the other amino acid. And in the sequence, it is written as a single letter code for the amino acid. Now, single letter codes are standard, defined already. If you go to the Google, just search for the amino acid symbol, you will get it, and they are pretty standard. When we write in a single format, they are called the protein sequences. There are many specific formats, but most widely accepted used format is called FASTA format. We'll talk about the FASTA format in a moment when I will show it to you. So that's a protein sequence as a primary structure. When we take additional information other than protein structure, so you have a protein structure, Though we call it as primary structure of the protein, it is actually not a structure. It is just a protein sequence. There's no structure. The structure is how the amino acids are connected to each other. That information is given to you. So that's a primary structure of the protein. The secondary structure has added information that what part of the protein sequence is made up of alpha helices and what part of the protein sequence is made up of beta strand. We do not have any information that how they are, how do they look like in the, how they are oriented in a three-dimensional space. It means that you have a protein linear sequence. One part of the protein sequence is a alpha helix, and then strand, then alpha helix, and strand, and so on, likewise. But you do not have whether they are alpha helix are together, two alpha helices are together. It's not. That information is present in the three-dimensional structure of the protein, where the sequence is oriented in the space. So you will understand which amino acid position is more closer to which amino acid position. It's not always linear. Because here, if you see the third amino acid here, third amino acid is here in the protein sequence form in this structure. Maybe this is the 50th amino acid which is nearby because it is in a coil shape, in a three-dimensional turn shape, right? And the same way, in the same way, when you have multiple sequences together, you form the quaternary structure of the protein. I think I have said enough. It's time to go and look at these two things one by one. Let me go to the database for protein and DNA. So if you are talking about the sequences of the DNA and protein, I have this one. If you have, okay, can I just do this one? I don't know how this will. Okay, now I go to this this one and say what? NCBI nucleotide. NCBI nucleotide. Okay, so NCBI nucleotide is one of the sequence database of DNA. We can say any protein name, any protein name where the DNA we need to identify, or any gene name we can say. We can say, for example, base is a Homo sapiens base 1. Let me see if there is any. So you can search by accession number, or you can search by any term. Whatever research project you have in mind or given it to you by your professor, you can look at those entries. You need to ensure that you select the nucleotide here, because there are many many other databases offered by the NCBI. You see the base 1 is a beta secretase 1 of Homo sapiens, which is a human. And you have a lot of information about this one, about this particular gene. The important information, all are important, but what I am talking about, the important information at this moment is the DNA, is a gene sequence. So here you see the interactions here. If you see gene information, a lot of information. I am scrolling a little fast, and you see the information. Yes, reviewed status. Okay, keep looking to the information, which is mRNA, protein accession. This is also linking to, so the other informations are also linked here. What you need to see is to search for a genomic sequence, FASTA, range from this to this, And you see, if I click on the FASTA, you will get the sequence of the DNA. So you have A, T, G, C in a specific order. What it says is it's a faster sequence of Homo sapiens, beta secretase 1, reference sequence gene, chromosome at chromosome 11. Now, the first line that you need to understand, there are multiple formats of representing the sequences of protein or DNA. This specific format is called FASTA format, where the sequence starts with a greater than sign. Whenever you see the sequence starting with greater than sign, first line is comment, whatever you want to write. You want to write your name, that's fine. Any experiment name you want to write, that's fine. It has no impact on the sequence itself. The characters of the sequence are one letter code for the adenine, thymine, guanine, and cytosine, which is repeated, uh, which is present in a specific order, and that information actually codes for the actual protein. In each line, there should be 60 characters, and there is no termination sign, for example, no end sign, for example, slash, comma, asterisk, nothing is there. So no gap in the sequence, all are continuous characters starting with greater than sign. That's a FASTA sequence format. All right, so we have seen FASTA sequence for the DNA, which is fine. Let me go and introduce you with another database, which is called UniProt. So UniProt is again another very important database, and I can go to the same protein that I have shown you in the in the nucleotide gene, which is base 1. I can go and search for base 1 here, base 1 for human. This one, base 1 is human, P56817. And if you remember, that was also there in a link. Just let me go down. P56817. You see, that's the same protein, which is, I have opened it here, search here. So base 1 is present in human, mouse. All are not same. They may be same, but they may be different. Many times they are different, slightly different. Even one amino acid different is a different. It can lead to a lot of difference. It depends where that difference actually present in the protein. So let me open this one. You see a lot of information. We'll go cover a lot of things in the upcoming sessions. But for your information at this moment, a lot of information is provided. You go to the sequence and isoform, and you see there is a sequence of the protein written. This is a very different format, not the FASTA, because it's not starting with greater than sign. It has gaps, it has numbers. So it is definitely not a FASTA. Let me open the FASTA one. So see this, when I clicked on download, it has now the FASTA formatted sequence. It starts with greater than sign. SP is just so it's prod, ID, description about the protein. The first line is complete. Now from the second line, it has one single letter code for the amino acids, which are methionine, alanine, glutamine, etc., etc., etc. Okay, so this is the protein sequence. So this is a primary structure of the protein. The structure is not there, but the structure in the form of a amino acid order, primary structure of the protein. Make sense? I show you another database which gives you a lot of other information. It's called Protein Data Bank. Now, Protein Data Bank has a lot of structures of the protein in a 3-dimensional. It means in a 3-dimensional space means either tertiary structure or quaternary structure. And then the accessory information of the secondary structure of the protein is also there. So let me open the same protein of the gene base 1 or the P56817 there for a for a for an ID W51. So let me open this one. I'm just looking for one W51, one particular entry in protein data bank, which should show me the structure, which is like this. And let me go to sequence information. Okay. Now, if you see the sequence information here, you see a lot of information about the protein family, which family it belongs to. It has the class, architecture, topology, homology, domain information. Yeah, it is showing on top of that. So the family is eukaryotic aspartyl protease. It is coming here. If you see my cursor and then look at those things on the top, you will see these things are changing. So you understand where exactly these things are showing. All right, there are other information thing that I wanted to show. Seems like they have changed a little bit things in the overall representation. Let me try it. Okay, no, I don't want this one in this way, certainly. See, okay, so they have changed the layout here. The layout is called secondary structure of the protein. So yellow is showing the sheet or strand information, and this pink is showing the helix, right? If you see this region here, it is showing, it is changing based my cursor position. If I'm mouse hovering on the yellow section, rectangle box or vertical boxes, it is showing the strand, a sheet, and this pink one are nothing but the helix. So what it is saying, if this is a sequence, which portion of the sequence is either helix or the beta strand or sheet? Now you can ask me, what happens to the things which are neither alpha helix nor beta strand or sheet? They are basically the loop region. The loop region means the connecting point between the two secondary structures. So for example, this helix is connecting to this helix by means of a loop. Okay. So that information is given by the blank. So primary structure is the sequence which is given here. Secondary structure is giving you the information of which part of the protein sequence is this alpha helix or beta strand. The tertiary structure of the protein is the three-dimensional structure of the arrangement of these secondary structure. Let me show it to you that also. But wait a moment. You need to know how to see those structures. And this is very important. You need to do a lot of hands-on just to get the full confidence or the control of the mouse and how the mouse button behaves, because there are multiple softwares open sub that structure. So I will go to the structure summary again for 1W51, which is here. And I can go to the download file, which is a PDB format file. But I will not do this for this particular protein. And I have a reason for it. I wanted to show you a protein or the entry which, which has both the things, protein as well as DNA. So, you know the proteins which binds to the DNA, ligases, transcription factors, helicases, these kind of proteins binds to the DNA, and people have identified their structures in a complex form. In a complex means they are where both the things are present, the DNA and the protein. So I have one entry for you. Let me go to that slide quickly. 1x9n. Okay, let me open that 1x9n for you. 1x9n. So crystal structure of human DNA ligase bound to 5-adenylated nicked DNA. The DNA is nicked, it's cutted. Okay, so what I will do, and you see there is a DNA, there's a protein. I will go to download file and I will go to and click the PDB format. That's it. This is what I need. But how do I open this file? I need a visualization that can generate this kind of file. We'll go to that files in little detail in a completely different session altogether, because there are a lot many details here which we cannot cover. The focus here is to understand, to let you understand the basics of structures and some bit of control on what you can see in the structure of DNA and protein, so that you can understand the later story of the program. So I have a program. There are so many softwares. We'll cover those things, but there's one important software which is freely available is ICM Browser. ICM Browser. So ICM is from Molsoft, it's a private company, and they provide a paid softwares, but browser is free of cost. So for studies purpose, for educational purpose, we can download it. I have downloaded for this program. If you see ICM Browser Pro is there. Let me open that one. You can simply go download, put the email ID and install it. It's a little heavy. It will take some time, but you can download it. So this is how the interface is. You have left-hand side control. You have some command line operations, menu button, toolbar options, and this gray big screen is for rendering the structure of the proteins or the biological macromolecules. So I will go to the file, open, file, open. What I want to open is the structure of the protein. 1x9, I believe. That structure should be there in the download. So download is here, 1x9n is here. Don't worry about this candle. We'll talk about this one later. And you see, I am loading the structure. Can anybody confirm me you are able to see that structure of this molecule?
Yes, we can see the molecule.
Perfect. Perfect. Very good. So I think you are. And what I'm doing is I'm left clicking my mouse, left button, and holding it and then moving the mouse. You see, I am clicking my left mouse button, holding it and moving it. Are you able to see protein and DNA together? Write in the chat box. Are you able to see protein and DNA in this rendering? Right. Are you able to see base pairing in this structure? Say yes or no.
Yes.
Very good. Are you able to see a DNA is cut? DNA has a breakage. Are you able to see? Let me move it a little bit more. Are you able to see any breakage in the DNA? You need to locate yourself. Keep your eyes at a place where you can see. Okay, very nice. Beautiful structure. Experimentally determined. Downloaded from protein data bank. DNA ligase protein working in a way to join the two ends of the nicked DNA by making a phosphodiester bond. Yes. On the left hand side, you see there are some chains. mm, so these are 3 DNA. That's why there are, you see, BCD, N for nucleotide, and A for amino acid. So the protein is single chain 632 amino acid. This is how you should read it. If I uncheck it, and you see this orange is orange. If I uncheck it, the protein will go away. You see, protein is gone. What you are seeing is the DNA, but I see DNA is just one. No, the DNA has 3 strands now. Because a double helical structure, 2 strands goes in parallel, but one strand in this particular structure is broken. That's why you have 3 strands. 3 strands. One strand, another nicked strand, and third is the one continuous strand, and that's why you have 3. The green one is T chain, you see. The other one is yellow is C chain, and B chain is this. That's why the whole structure is formed in this particular entry. But you can ask me, this pairing is not the base pairing, Puneet, you told me or showed me in the presentation. Is it something wrong with the structure? No, actually not. It's just representation of the protein and the DNA to make your life easy to understand. We can change that way. For example, let me take out, remove this one and say this is X stick representation. So you have the stick representation of the base pairs or bases. Here also I can make X stick so that you can. Now you are saying, Puneet, it's too crowded. I can't understand the base pairing and so whatever. Can you make it more clear for me? So, okay, what we can do is we can remove the ribbon representation so that it becomes little clear. No, remove the other ribbon as well. Okay, let's do that word. Okay, now I see. Are you able, let me remove the fog. Are you able, okay, let me do, this white is too bright. Let me do a change in the color. Are you able to see it now? Little clear. Ball and stick representation of the bases in a base pair form so that you can understand it's a double helical structure. What we are talking about the DNA. What we are talking about the DNA. Okay, that's good. But Puneet, one thing is missing. The one thing which is missing is the interaction, which is not shown here between the base pairs. For this, I need to do some magic. The magic here, I need to add hydrogens and prepare it for it. So I will do the right click on the first form where the protein entry is written 1x9n and say convert PDB so that it can do a lot of things. Don't worry about it. Optimizing hydrogens and adding hydrogen is something which is critical here. And let me do this. So it is doing this job automatically. So very beautiful software. Okay, still doing. Is it doing only for this? These 2 visible entries? No, it is doing for everything, for all the amino acid, for all the DNA, etc. Okay, too complex. Let me remove all these because it has created a clone after processed thing. So I will remove the protein for the sake of clarity, making the DNA as X-stick, and you see the hydrogens as white are added. So each atom has a different color code. Oxygen is red, carbon is green here, and nitrogens are N, the blue one. Here also I can do X-stick. Here also I can do X-stick. And what else I can do is to remove the ribbon. And here also I can remove the ribbon. Here also I can do. Okay, this is fine. Let me do one more thing, just to view. Are you able to see the hydrogens here? Hydrogen bonds should be here. Labels, display. Okay, display hydrogen. You see this button? Let me click here and you see what happens in the main screen, right? Let me remove it, some bit of it, so that you can have even clear picture. See, okay. Now you see hydrogen bonds are there. Adenine, thymine, 2, cytosine, guanine. Some are small, some are thick. This is the distance between both of them. All right. Now, are you able to see the hydrogen bonds between the bases? Are you seeing the DNA is connected in a single strand with their backbone, the phosphate? You are able to see the pairing between the uh, between the bases. All right, you are seeing a lot of things. Believe me, you are seeing a lot of things. But you say, okay, this is good for DNA, but what about this protein? I am so much interested about the protein. Okay, let me see the protein part as well. You see, this is the whole protein. I can make it more view F11 for full screen, so that you can have really wonderful picture. And you see the amino acids are written like are displayed like this. Some of them are only shown for sake of clarity. If I make everything ball and stick, it becomes too heavy for you. Let me do that. It becomes too heavy for you. You see this one, right? Maybe not able to understand. Okay, let me zoom in a little bit more. Oh, it's still crowded, right? Okay, let me see this one. If I proline, serine, you see, I select only 3 amino acids, and which are here. As I add more amino acid, they will keep on connecting with each other, right? They all are connected. I am doing the central click and then translating it. You see, they are connected with the backbone. CONH bond, CONH bond, CONH bond, then C alpha, CONH, CONH, C alpha, and this is what you have studied in your uh, in your college days textbooks. Okay, so amino acids. This is a three-dimensional structure, and now you see three-dimensional structure is important because these two amino acids you see, they are of different number. Let me label them. Display residues. Okay, you see tyrosine 266 and tyrosine 273. These two amino acids are basically distant amino acid, 66 and 73. There's not 66 and 67, but still they are closer to each other because of the three-dimensional twist. And, and that's why three-dimensional structure understanding is important, which this information cannot be leveraged just by having a primary structure of the protein and secondary structure of the protein. I hope I'm able to clarify this to you. and it is making some sense to you. Let me go back to the presentation. Okay, so we said that structure, now you have more clarity on that part, which is good. Let me go to the another section. We said that, okay, you mentioned about the protein structure in good detail, and protein sequences are also seen. But in case you have multiple sequences and you want to see both of them, all the sequences in one view, which program should we use? And there's a lot of analysis actually done on the protein sequences. JALView is one of the great application, I would say, freely available, absolutely amazing application to visualize the sequences as well as the structures of the protein. We'll go to that part in a while. So what I'm doing is, we are doing just one exercise here. Sorry. We'll go to the UniProt and we'll say, okay, UniProt, tell me all the protein sequences of aspartic proteases. Aspartic proteases. So I search for aspartic proteases, you see 546,000 results. Can you believe it? It's just too much. But we should not download all of them and do anything. We'll take out some organism we can select. For example, in human we can select. In human, still there are 114. Just to make them simple or easy or small, we will say show only with 5 annotation score, means they are very well curated, identified, very well reported. And I can do what? I can do download. I'm saying canonical is nothing but the representative, because each protein can have several variants, and that's a representative one. I'm not saying I need the isoform one. I don't need any other format because I know FASTA format very well. It starts with greater than sign and it has a comment. It has a single letter, a minus it from the second line. No gap, no number, no sign of termination, 60 character in each line. So I know what the FASTA format is. So I will do that. I do not want to uncompress it. That's why I'm saying no compression, and I'm downloading it. So this one I downloaded. Okay. I go to JALView. JALView I installed from the JALView website, that I can also show it to you. Go to JALView. Very, very beautiful website, very beautiful tool. You see, JALView, a lot of information about it, and you really enjoy doing this. If your work, thesis work involves multiple protein sequences, DNA sequence to be analyzed, understand, and do a lot of analysis, certainly this can be a very good tool. You see, protein data bank is written, structure, you can do alignment, you can generate these kind of things, and you have seen these kind of things in publications multiple times, and you see the clustering is also done. Which protein is related to which protein in a closest form, or distantly related. So okay, having said that, I downloaded this one. I have this one installed. I can remove this one, which is good. No sample is required here. I can go to the input alignment from file, though it's not aligned one, but I can go with the sequences. Oh, there are 3 1. Why? There should be more. Let me open file. Oh, which one is the latest one? I did a lot of downloading. 28 1, 13 52. I believe this one. No, this is, how do I identify? There's no details. Let me see details. Which one is downloaded? 722. Okay. 720. 832. 832. This one. Yes, this one should be the protein. Yeah, you have all the proteins here. All right, all right, all right. So I have all the proteins here, but they are not aligned. What we can do, go to the web services, alignment. There are so many functions. I'm doing it just for reset, quick alignment. It is doing quick alignment for us. You see, alignment will be done in short while. You will have the amino acid aligned. They are just arranged one below the other, but they are not aligned. You see the alignment is there. Oh, just very less aligned proteins because they are very different, I believe, proteins. Let me go to the color code and say percentage INT. Oh, yes, you have leucine here in this particular column. It seems like leucine is a very important critical amino acid for aspartic proteases here. Okay, good. What else? Let me go here and see, is there anything other? Oh, yes, you have other amino acid also very much conserved, but very less conservation among the proteins, which is fine. It's just fine. Okay, going a little fast. There are other amino acid. Okay, yeah, good. Couple of amino acids which are present here. I don't think there is a big line with all the amino acids very well conserved. So what I will do, I will say blossom score. Okay. So different way you can play around it. You have proteins. If you double click on this, you will open the particular protein entry page. You can also embed the structure of the protein here, which is in the sequence. We'll talk about a little bit more with some functions of this particular tool. But for sequences analysis, this is the great tool. Okay, which is fine. We learned it. It was good. Let me go again to a particular, to our presentation. JAL view, we have done this one. ICM Pro, quickly 1x9n, we have seen it. Now, we talked about cell, intracellular environment, structure, extracellular environment, space. I have a picture here which says about the therapeutic strategies for the COVID-19 treatment. What you are seeing on the image, there are two different strategies. One is immunomodulatory kind of approaches, inflammatory kind of things, and second is the which has the information about the viral replication. Okay, now you see this particular portion, which is in the light pink here. You say host cell, and you can identify what it is. Can you identify? This is a nucleus. This is the endoplasmic reticulum. This is a Golgi body. And this is what it is where the things are reported. These are nothing but the cytoplasm. Now you can connect what people are talking about. There are drugs mentioned for each of these strategies here. It means that people are talking about having a medicine which can act on the portion of the or target to a place in the cytoplasm or in the extracellular environment on the on the cellular membrane and treat the disease, treat the patient and cure the disease. This is a virus which has 4 different things: spike protein, envelope protein, membrane protein, nucleocapsid protein. Protein, right? Protein. You understand protein. You understand what we are talking about as a cell. You understand this is cell environment. We already talked about medicine has to reach to the place where it should go and bind to certain biological macromolecule. These are membranes bound protein where 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 virus binds to it and then go for endocytosis. It uncoats, it releases its RNA, then do the replication of RNA, make multiple copies. It produces a new forms of multiple viruses, and then it release out. Different places, you have different strategies. You can block the entry of the virus by means of several drugs. You can act on the RNA replication part or translation part of the anti-RNA polymerases as a drug. You can act on the protease inhibitor so that this virus factory can be taken into consideration, and there are medicine for it. There are other strategies which can block the release of this particular assembly of the viruses by using antiviral release strategy, right? On the right hand side, you have other inflammatory cytokines related approaches to treat the patient. Point is that there could be multiple things, but the fundamental is where your molecule is sitting. What is your molecule? How your drug will interact to the molecule. And that is all we will talk about in the coming sessions. I hope with this one, you have certain understanding of at a macro level what we are talking about, what we are looking into it, how we should look into it. Now you are prepared for a deep dive in the upcoming sessions. We also mentioned about a protein-protein interaction, which is a string database, and I can give you a very simple glimpse of how it looks like. When two proteins interact with each other, you need that information in hand so that you can block that, or you can use this as a machinery for identification of the drugs. I can go to the string DB, string database, and then you see this is a protein name. I can say base one, the same protein I can take. Auto detect the organism, we need only the human one. Okay, base one, maybe it is taking in a different way. So let's say base. So it's saying beta site of APP cleaving enzyme, which is fine. I am interested in Homo sapiens. Let me continue. And is generating the protein-protein interaction. Means that a protein called base in the human cells, wherever it is located, it interacts with so many other proteins. These are the proteins. So if I need to design the drug for the base one, I need to consider the artifact that it also stop interacting. It may stop, it may interfere in making the interaction with the base one to other proteins, and that can cause the problem. All those things needs to be considered while taking the consideration of the drug target into the mind. We'll talk about it a bit more on drug targets and what are the characteristics of the drug target should have while selecting a particular drug target for the treatment, for the designing of the molecules and treatment of the diseases. Having said that, I end the session with this particular note. I believe you will do the hands-on exercise at your place. Download, install JALView. You should be able to search sequences using UniProt and nucleotide databases. You will download them, open them, analyze them a little bit, and you should be able to download the structure of the proteins or DNA from Protein Data Bank, install Microsoft ICM Browser, and do hands-on exercise. Let me know if you have any questions. I see a lot many chat points are there. Thank you very much.
Thank you so much, sir, for that wonderful presentation. And we have one query from Abhimanyu Avasti. He's asking, is there a database about pathological pathways about disease?
Yes, there are multiple databases for the pathways. cake is one pathway, reactome is another pathway, and they are indeed little complex, but fundamental for the pathway is one after the other connections. These connections are nothing but the connections of the proteins and other biological macromolecules. So I believe you should be able to explore the pathways. If you as you search for reactome or cake pathways or wiki pathways, you should be able to search them effectively.
Thank you, sir. And we have Saptarshi asking what is Caver software tool?
What is that?
Caver, CAVER software tool.
Um, CAV, Java software. Okay, I am not aware of. There are so many software. Let me see. Java, is it for identification of tunnels and channels? Is it? Yeah, there are. So there is a software for protein analysis and visualization. It's called Caver, and there are other variants of it. I am seeing this into the screen. It seems like gold standard for analysis of tunnels and channels in biomolecule. Okay, so there are molecular macromolecules, biological macromolecules, which makes the tunnels or ion channels. This particular software is, as they claim, is very very efficient analyzing those tunnels and channels of protein structures. Go ahead, explore that software. I will also do that. It's a good information for me as well. So there must be, I believe, certain options which helps you to calculate the internal structure, the volume available in the channels and tunnels, so that you can have some designing opportunities. You can design the drugs accordingly. So that's very good. If that is available free of cost, why not explore that?
Thank you so much, sir. So I guess that was the end of the questions. So I can see that a lot of the participants are yet to enroll for the program. So for such participants, we would like to share the registration and program details. So for that, I'd like to pass on the stage to Shubham, who will be explaining more about program registration. Over to you, Shubham.
Thank you, Shigori. So let me first share my screen. Yeah.
Yes, Shubham, we can see the program page.
Yeah. So hello everyone. I'm Shubham. I'm the Business Development and Marketing Associate here at Pine Biotech. So I'll be briefing you about the registration part for those who haven't enrolled yet. So as you can see, this is our program page. So I'm pasting the link so that you can also go through this link after the session. And let us, okay. So as you can see, this is a program page for Cheminformatics for Biomedical Drug Discovery, and it has it has been commenced from today. So yeah, when you will scroll down a bit, you will see there's a program overview. In this section, we have summarized what all the topics or the concepts you will be learning from this program, as well as how you can apply this concepts or the topics for a research project if you are from a biotechnology background or a pharma or any other industry. So you can apply this knowledge. So when you will scroll down more, you can see that there is an interactive session calendar here and some of the dates are highlighted. So like today on 28th Jan, we have a first session for the Cheminformatics and we have learned about the computational structure biology and some of the topics you can see from here, which we have covered. And yes, and the next session we will be doing tomorrow, that is 29th, and the topics which we will be covering you can see on the right side of this page. Now coming down a bit more, you can see that we have three different tenures for this program. One is for 45 days, 60 days, and 90 days. So the common resources we have for all these tenures is you will get the online tutorials, then you will go through, like you will have the access of the Omics Logic Learn platform where you can go through the project examples related to this particular program. Along with that you will get a coding session, and by chance if you miss any of the session, then we will provide you with the session recordings. You will get the online mentor and technical support, and after the completion of this program definitely you will get a certificate of completion. The extra thing what you will get in the 60 days is you will get the one-on-one mentor session, and let's say you have a research proposal related to this and you want to like work on that thing, then you can draft your research proposal also, as well as you can make it a poster, and we have that symposium, then you can present in a poster presentation. And for the 90 days, apart from all this, you will get a specific pipeline on for the TBA and for server, a one-on-one mentor session, and you can independently work on your research project and get it published also in the peer-reviewed journal part. So these are the different tenures and the resources with their outcomes. Coming to the pricing part, as we can see on the screen, this is for in USD. So for 45 days it is $150, for 60 days it is $300, and for 90 days it is $450. Let's say some of them are from India. So what you have to just do is you will see a drop down option here. So you have to click on that and select the currency. So you can like for the 45 days in INR we have around 7500. Then for the 60 days it is 9999, and for 90 days it is 14999. And similar for the our African community, just click on the drop down and you can click on this Naira for the Nigerian ones, and you can see the pricing here as well. Now how we can register or how we can enroll for this particular program. So let's say I'm interested in 90 days program, and simply just you have to click on buy now, and after that you will see the checkout page, and just you have to fill your user details and click on this checkbox, and you can select a payment method, either Stripe or PayPal. If you're facing any issue in both these payment, then we have alternatives also via PayThen and via for Indians UPay option is also there. So we have options for alternative like registrations. So this is all about the registration part. So if you have any query, please feel free so that I can address those things.
Hello, can you hear me?
Hello.
Hello, yeah, can you hear me? I'm Oluwasegun from Nigeria.
Yeah. Hello. Hi, once again.
Yeah, I just have a brief question. I enrolled for the Cheminformatics program, and I'm finding it I really can't. I'm not able to locate the course on the portal. Is it that I will search for the chemoinformatics topic on the search bar, then the courses under it? Those are the courses I'm going to be working on, who I'm working with.
Uh, like like to be precise, like what I have understood is you want to work on the research part, right? Related to the
Yes,
chem
yes,
informatics. Yes,
yes,
of
yes.
course. Yeah, of course you can work. Like we will be having live sessions with Dr. Puneet, and after
Okay.
that, if you come up with a research question or a research proposal and you want to work
No,
on that thing,
um,
that is also
so
possible.
no, you what I'm trying to say is I've not been able to access the courses on my portal.
So you can just mail so that we can verify with her team. Like there might be some technical issue. So Shigori has shared
Okay.
the contact details. So please mail us at that particular ID so that we can see what issue
Okay.
you are
Okay. No
facing.
problem. Okay. No
Uh,
problem. Thank you.
Thank you.
All right. Thank you
So
so much. And does any participants have any further queries? If so, please feel free to share it in the chat and we'll be happy to answer. All right. If the participants have no further queries, along the way, if you're having any questions or concerns, you can reach out to us at the email ID and the contact number that I've provided in the chat. So please save it and we'll get back to you for any questions or concerns. And with that, thank you everyone for joining today's session. And like Shubham said, we have our next session tomorrow. So we look forward to seeing you for that as well. So with that, thank you everyone and have a great day.
thank you everyone.